DNA and RNA, nucleic acids. Okay, oh, this is nasty. All right. Where did they find these? Discovered the molecules and isolated them in 1869. Okay, think about this. 1869. Where did they look? The pus cells of discarded surgical bandages in 1869. This does not sound like a good job for anyone, okay? And probably a grad student, not the guy who got his name associated with his work. Or going out there collecting this stuff, all right? And they were molecules, and they were called at the time, or they identified them as DNA deoxyribonucleic acids. Now, organic structure. Quite frankly, this was kind of more of an organic identification of a molecule. They didn't know what it did, and didn't know what it did for a very long time. But now that we understand what DNA is, we can understand why the components of the DNA and RNA are important. The DNA RNA is composed of a base, a sugar, and a phosphate group. Okay. Why in the world is that ring structure a base? Why do we call it a base? It's too simple a question for you to really think that hard about it. Yes? Because it has nitrogens. That's exactly it. All right? And so that's why we call it a base pair. It's like ammonia in terms of a base. The ring structure is based upon ribose. Ribose is actually a derivative of glucose. And our body converts glucose into ribose. Now, we normally think of glucose being broken down by a pathway called, does anybody remember? What breaks glucose down? What? Glycolysis, okay? Which then you might have remembered feeds into the citric acid cycle, which then the molecules feed into oxidative phosphorylation. That's what the traditional pathway we think about, but sugar goes in a lot of different ways, and we will learn about what's called a pentose phosphate pathway, in which we are going to make these ribose molecules. Ribose are important sugars. And then we add phosphates onto them. If it is a base pair and a sugar, it's called a nucleoside. If it has a base pair, a sugar, and at least one phosphate, it's called a nucleotide. Okay? And we see most often represented nucleotide, a triphosphate. And the one that we reference most often is ATP, although that's not the only one. Okay. The base pairs will carry the genetic information in terms of the linear sequence of how they're connected. The sugar and the phosphates create the structural integrity that hold the DNA together. DNA itself is pictured below in the beta position and the 2 position. Instead of a hydroxide, there's a proton. And that is where we get the deoxy, the loss of the oxygen. And what that invokes is a lot more stability in the structural component. The bases are either one of 2 types, a purine, a pyrimidine. Purine, small name, big structure. Pyrimidine, big name, small ring. Okay? Now, the attachment is important. The 9th position nitrogen on the purine is attached to the 1 position on the sugar, and it is always beta or above the ring. And then the 1 position nitrogen of the pyrimidine is attached to the 1 carbon of the sugar in the beta linkage or above the ring. These you should know, highlight, star, memorize, put into your heart of hearts. Okay? The purines and the pyrimidines. We have 2 purines, we have 3 pyrimidines. The thymine and the uracil are exclusive. Thymine is in DNA, uracil is in RNA. The cytosine, the adenine and guanine are interchangeable between the 2. Those you need to memorize, those 5 structures. Now, here's the deal about DNA and RNA. DNA is pretty good about using just those 4 base pairs that it uses. RNA, not so much. RNA has a lot of unusual base pairs. All right. Now, these base pairs are not random. They are actually important, and you see them most often in RNA that contains structure, okay, and function. And so you will see these unusual base pairs in, let's say, versions of RNA like transfer RNA and ribosomal RNA, of which some of these confer function. Now, do you have to memorize all of these? Absolutely not. What I do want you to know is that there are unusual amino acids, base pairs, and when we get to those structures, you'll see different designates, single letter representatives, where we think of A, G, C, T, right, or U as single letter. We will have other letters or even Greek symbols to represent some of these. But we'll introduce those in terms of function. I just wanted you to know they exist and they're important. Nucleoside and nucleotide, I have no idea why I taught that twice. So it must be important if I taught it twice. DNA structure is linked, okay, and there's a directionality. When we talk about a sequence of DNA or RNA, the beginning we find is what's called the 5' hydroxide. That's the start of it. The linkage between base pairs or the sugars is between the 3' hydroxide and the 5' hydroxide of the next base, and is linked through a phosphodiester bond. All right. So what that does, it means that between every single base there's a phosphate. What charge is the phosphate? What charge is the DNA then? It is very, very negative, okay? And that's very important. Now, we can note the DNA structure, and we can say there's a phosphate and then a base pair. And there's a phosphate and then a base pair. And there's a phosphate and there's a base pair. And a phosphate and there's a base pair. But we are scientists and we're lazy, and since we know there's a base pair between them, we very rarely ever put that. And so we can just single out the base pairs, okay? And if written, the assumption is the first base pair is 5' and the last base pair is 3' unless otherwise designated. So you can put 3' to 5', you write 3' at the front, 5' at the bottom, and reverse it, but you have to tell people that. If you just write the letters, you assume 5' is the first. In this, even when we designate them, what we did not show is that there is a ribose there, but a ribose is part of the structure, and so even when we designate the phosphates, we still were leaving out that there were ribose structures. All right, so how in the world did we discover what DNA did? So 1869, we identified the organic structure of the nucleic acids. Not until 1928 did we kind of get an idea or had an experiment that supported the idea of what it might do. So that's a lot of years of not knowing what's going on. Almost 60 years. Okay? And so what we're going to use is experiments with the bacterium that causes pneumonia. Okay? And there are 2 versions, the S version and the R version. And it's called encapsulated or non-encapsulated. The bacteria that is encapsulated has around it surrounding a polysaccharide coat. Okay, so it must be cold. It needs a coat. All right, what in the world is a polysaccharide coat? Sugars. Okay, so it's a linked set of sugars that surround it. Okay. Now, that sugars on the outside of the bacterium is what makes it infectious, because the cells will bind to that sugar. Okay, remember we saw a molecule that binds sugars that were called selectins. Okay, that recognizes the sugar and it brings it in. Oh, but it brings in a bacteria that is not there for anybody's good. And we now know when that is ingested, the DNA is incorporated, you get infected, and you have to die. The non-encapsulated is the bacterium that does not have the polysaccharides, so it is not pathogenic because there is no binding to a cell that's recognized and it doesn't take it in. Okay, so S and R version. S bad, R good. All right, we don't do these kinds of experiments too often anymore. PETA doesn't let us, okay, as much as once was, but here we go. Live, encapsulated, virulent bacteria, the S version. We inject it, and what happens to the mouse? It dies. Okay? All right, so we know this is bad. We take the R version, we inject it into mice, and it lives. Okay? So, between the two, there's something that is killing the mice. We're trying to identify what it is. Okay? And so this is something we also did. We took the live encapsulated virulent bacteria and we heated it. We took that heated mixture and we injected the mouse, and the mouse lives. All right. And so inside the cell there are basically three kinds of molecules that are important. One, the DNA RNA. Okay, we didn't know what that did. Two, proteins. And three, sugars. Okay? We're trying to figure out how biologically all of them work. All right, we now understand the sugars are what binds to the cells and allow it to come in. What happens from your understanding here? Why did the deadly bacterium become non-deadly when we heated it?
He broke down that.
Heat broke down what?
The coat around it.
Okay, that's one possibility. Heat broke down the coat. What else? What is another possibility?
Rearrangement.
Heat broke down the DNA. That's another possibility. What's the third possibility?
Dissolve the sugars.
Hmm?
Could have dissolved the sugars.
Dissolve the sugars. Okay. Anything else?
Denatured proteins.
Denatured proteins. Okay. So those are all three possibilities. Now, melting the sugars. Okay, that would be good if they were mono or disaccharides. The fact that they are covalently linked doesn't let them be dissolved anything more. Okay, the heat was not big enough to break down the sugars coat because the covalent bonds are pretty tough in terms of their stability. Okay, not hot enough to break down the DNA. Okay, but what it is going to do is it's going to denature the proteins, means unfold them where they're not functional. So, yes, you've got to get the DNA in, but proteins have to work to integrate the DNA into the host for it to work. Now, we didn't know this at this time. Okay, this is from your knowledge now. Okay, but what they did know was the heat did something there that made it inactive. Okay. Now, what they did was they took the heat killed, we said doesn't kill mouse, and we took the R version, doesn't kill mouse, and we mixed them together. And the mixture of two species that don't kill the mouse kills the mouse. Okay. So what was proposed was there was something from the S version that was heated that was transferred into the R version that made it deadly. That's what they concluded. And so they tried to isolate different pieces of the puzzle. They isolated the sugars from the heat-killed version and mixed it with R. They isolated the proteins from the heat-killed version and mixed it with the R. And they isolated the nucleic acids from the heat-killed version and mixed it. Only one of them caused something to happen. When they isolated the molecules that were the DNA from the heat-killed with the R, it created the deadly version. Proteins didn't, and the sugars didn't. Now, what they called this was the transforming principle. Okay, that DNA, because it transformed a non-virulent version into a virulent. What happened was it transferred the segment of DNA that caused the synthesis of the saccharide coat to be made. Okay? And so here's all the words that we did with what we saw with the pictures. Okay? The transforming principle, okay, turned out to be that DNA. In terms of the extracted from the S version was found to contain the material possessing the most likely the property of DNA. Okay, and so this was the idea that DNA could create or transfer informational and functional relationship from one entity to another. One of the first experiments that supported that idea. Okay. Now, DNA, again, was in the cell, so they started to really study it. In the 40s, Chargaff determined that the base composition of DNA varies between one species and another. Okay? So you take a rabbit and you take a horse, and the composition, the percentages of DNA of A's and T's and G's and C's were absolutely different. Okay? But what he did find was this. You take a little tiny rabbit or a very old rabbit, you take a very fat rabbit, and you took a starved rabbit, and when you did and looked at their DNA, the percentage of A's, G's, C's, and T's never changed. Now, their composition, how much fat they had, how much protein, what was functioning, all sorts of things changed between their life cycle, but the percentages of DNA never changed within a species. That was kind of interesting. Had no idea what was going on there, but it was something that he looked at. He also came up with a rule. He found that when he calculated the percentages, the amount of A's plus the amount of G's is equal to the amount of T's plus the amount of C's. Okay? And that was kind of weird. A and G and B and C. Now, why does that work now? Now that you know something. Why in the world does A plus G equals T plus C in their DNA? Oh, you know this. Yes. Every A in the DNA must bind to a what? T. And every G must bind to a C. And so if you have A and G, you also have a C and T. And so that's why there's percentages. Now, he didn't know this at the time. He just found this to be kind of odd, and it was consistent. And so as we developed our understanding of biology, sometimes we find a very odd thing that happens all the time, and we don't know why, but it creates a body of knowledge that after a long period of time, we can write a chapter in the book, and it looks like it all went together. But this guy was working all by himself and got all of this information, and then somebody else read it, and oh, it works together. And so that's kind of how science works sometimes. Okay. The DNA double helix kind of explained to Chargaff why his mathematics work, and this was credited to Watson and Crick about the deduced three-dimensional structure of DNA in 1953. But it was from X-ray analysis conducted by Miss Franklin here, where, quite frankly, she probably should have also won the Nobel, really because of her work and her crystal structure and her analysis. You actually look in her notebook, she came up with the idea of the double helix before Watson Crick. Unfortunately, she passed before the awards committee awarded it, and they did not give Nobels to people who have passed. The two chains in this double helical nature that you probably have seen since elementary school contains a coil around a common axis. We call it the helix. And the DNA backbone are held together by hydrogen bonds between A's and T's. And so you see some structures of DNA that have been crystallized. Okay? There's an A form, there's a B form, and there's a Z form. Which form is the most often, which is the normal? The B form. Why in the world do we have A and Z form there?
Different conditions.
Different conditions. A crystal structure, these kinds of structures that we see, wonderfully detailed. Absolutely wonderfully detailed. But the problem is, it is not biologically the conditions in which you find things. To create a crystal structure, we have to have really unusual conditions, very, very concentrated molecules that make crystal structures rather than being a solution molecule that's a very less concentrated solution that's moving around. And so crystal structures, although give us wonderful data, sometimes give us data about a structure that doesn't represent what's in the cell itself. The Z form actually has the helix going in the wrong direction and not biologically active. The beta form is probably what we understand being the most biologically active. We have something called the major and the minor groove, okay? In that as we see this corkscrew or like the thread of a screw as you go up, you have something that this big bend here, major groove, this little one here, minor groove. These grooves are important because now we know proteins recognize either major or minor grooves and bind to them specifically. And what base pairs are there can make binding pockets that are unique. Depending upon which version of the DNA, you can get different, slightly different measurements. The base pairing, the distance between them at being 3.4 angstroms, is based upon the beta form, and it takes 10 base pairs to make one full turn of the helix. One interesting fact about the DNA is that it is anti-parallel. See at the top, one chain starts with 5', the other one starts with 3', and this orientation allows the correct hydrogen bonding between the A and the T's, and the G's and the C's. All right. Now, the fact that G's only bind to C's and A only binds to T's allows the idea of a complement to be formed, and the idea or the basic idea of transfer. If I split them apart, I can make a new strand that's exactly the copy or complement of what the original was. And so the whole idea about how genetic information flows or is copied comes from this pairing idea.